Alright, welcome back to session 4 of Outbreak Response. So in session 3, where we left it was we had extracted our protein of interest, so our potential antigen, and we'd run them out on a page gel to resolve the proteins and see what was in our mix. Did we actually successfully purify our protein or not? And we began the immunoblot. So why are we doing that immunoblot, or Western blot, as it's known by another name? So although a page gel, is very informative, it can't tell us whether or not the protein is immunogenic. It can't tell us anything about the physical antigenicity of that protein and whether or not it's going to be recognized within a human population as a vaccine. So we use a Western blot for that. And a Western blot will identify antibody-protein interactions, but only if they're specific. The anti-sera that we're going to use in this experiment, that is, what is our antibody solution, will represent a human population that has been infected with Streptococcus pneumoniae. Now, within that complex mix of antibodies, there may or may not be antibodies that will recognize and bind to our two antigens that we have expressed and purified. So that's why we're doing the immunoblot. And at the end of the last session, what we had done was simply take our page gel with all of our proteins resolved vertically, and then taken the gel and horizontally transferred our proteins onto a nitrocellulose membrane. It's that membrane that we're going to take today and finish off the Western blot. It's to the membrane that we will start to add our antibody solutions. Okay? And in that way, we will detect or not whether there are immunogenic proteins within our purified collection. Right, let's have a look. So let's have a look at this gel. This gel image is not exactly like the one that has been performed in this practical class, yet it does contain all of the same fractions or the same samples that we prepared within this practical. Starting off with our protein ladder, very important, so that we can look at the molecular weight of our protein of interest. And you can see that here in lane 1. In lane 2, we have our clarified protein sample, or clarified supernatant. You'll see that it's a complex mix of proteins, many proteins within that. Here is the unbound protein fraction, or the unbound fraction. So what we'll notice about this, which is special, is that there's one particular band which is missing from this lane. Right? What is the band that's missing? Well, it's clearly this one has been extracted unbound, meaning these are the proteins that remain after binding to IMAC, or the talon resin. And so we're demonstrating by running this fraction that a particular protein has been pulled out from the complex mix of the unbound, clarified supernatant. The next is a wash, just to demonstrate that there's nothing else coming off the resin and to clean the resin before elution. And then we have our elution or eluted protein. And this protein is larger than the protein PPSA or PLY that we're seeing in this prac. So that's what page will look like. This is an example of the results which are achieved within the prac. From this, we'll take that and transfer to nitrocellulose, and you'll see that done in the video that accompanies this week's session. Now back to this slide as I showed you in the last week's session. However, today in task 4.1, we're completing this Western blot. And that is, we're doing that which is above the membrane now. So at week 3, you remember we did this. We had the membrane with the antigen attached. We transferred all proteins to the membrane, and it's irreversibly stuck to nitrocellulose. In this week, we're going to start using our antibody solutions, primary antibody, or the patient's sera, which hopefully will seek out and bind to one of their antigens of interest in a specific fashion. We can't see that, however. There's no way to visualize that primary antibody in the format that it is. So we will then come on with a secondary antibody. And our secondary antibody has an enzyme which will be familiar to you, known as HRP, or horseradish peroxidase. It's the same enzyme that we used in the RPA naked eye detection that you remember from the first week. A secondary antibody is specific for the first antibody. It is an anti-human secondary antibody, meaning that it will bind to any human antibodies that it finds on the membrane. Now, hopefully there's very few of those that are only stuck to the proteins that we're actually looking for. We can detect now the presence of the secondary antibody, which in turn is detecting the first antibody, which in turn is detecting the antigen that we put onto the membranes through the use of the enzyme, the HRP. And we'll add a substrate to the membrane, as you'll see in the video, which allows for a color development. And it will turn a purple to black color on the membrane, and we should be able to see with the naked eye again the presence or absence of any particular banding patterns that might exist. Finally, I've stated that the protein is transferred to nitrocellulose. This is important. There are different membranes that you can transfer for Western blots, and we use nitrocellulose. That was done in week 3, and now we're moving into the next session. One question I have for you, though, is we blocked that membrane between session 3 and 4. Why? Why are we blocking that membrane? Okay, I'll hope that you'll be able to answer that in the week to come. Antibodies used, we're using an antibody sample, it was a human serum, which is from pneumococcus infected patients, as I've already said. So it should be able to detect pneumococcus specific proteins. And that brings us to the end of this practical. Okay, thank you, and we'll see you again for practical 2.